Hello there, my name is Evan Marth. I'm a biology major with a minor in mathematics at Athens State University in Athens, Alabama. I am working in the Carmichael lab with Dr. Cloyed, and my summer research project is to examine stable isotope values in fish, plankton, and sediments in the Mississippi Sound. With the use of stable isotopes, we can get an idea of the amount of nutrients that flow into the Gulf of Mexico from the rivers and watersheds that input fresh water into the Gulf. Stable isotopes can also help us understand food webs and trophic levels in an ecosystem. Without the flow of nutrients into an ecosystem, said ecosystem cannot maintain equilibrium to support the diversity living within it. On the other hand, too much nutrients could harm the diversity in an area. For example, if nitrogen-rich fresh water flowed into the Gulf, it could cause an algae bloom, and this bloom can result in eutrophication and kill large amounts of the fish species living in the Gulf of Mexico. First, we use a YSI device to take the temperature, salinity, and DO, or dissolved oxygen, of the water at the site. Then we use a horizontal water sampler to gather the water from two different depths to use later. After that, we use a sediment grabber to grab sediments from the seafloor. Once we get back to the lab, we will filter the water we have sampled. We will use a small motor that is attached to several tubes, and those are attached to flasks. When we start the filtering process, the motor creates a vacuum in the flask, pulling water through a small filter that is filtering out the plankton that we need. To obtain the fish we need, we use gill nets and trawls to collect the fish. Once we have the fish, I take them back to the lab to dissect them and collect muscle tissue samples via biopsy. These tissue samples will go into an oven for 2 days. After 2 days, they will be taken out, ground up with a mortar and pestle, transferred into a scent vial, and from there, they will be weighed and packed using an ultra microbalance scale. The packed samples will be sent to a lab to be analyzed using a mass spectrometer, and once analyzed, the data will be sent back to us, and from there, we'll be able to use it. We will also be sending off the water samples and sediment samples to the same lab to acquire the data we need for more statistical analysis.